right, good afternoon. Thank you for the opportunity to speak here today. My name is Lisa Murray. I'm Director of Operations at Provenir. We are a consulting firm that supports both new and generic drug development. And I know a number of the presentations this morning mentioned BCS biowaivers, and this is an established regulatory pathway that's been in existence for almost 20 years, but remains largely underutilized. And so today I'd like to explore and identify opportunities for extending applicability of BCS biowaivers, spreading awareness, and facilitating overall acceptance of this technique. As a disclaimer, I am a former employee of Absorption Systems, so some of the data that I'll be featuring is from Absorption Systems. Dr. Sid Bhupathi, who presented this morning, was kind enough to allow me to share some of this data. So BCS, Biopharmaceutics Classification System, is a regulatory framework that classifies compounds according to their aqueous solubility and intestinal permeability. According to the FDA BCS guidance, which was issued in 1996 and revised in 2000, biowaivers are eligible for immediate release solid oral dosage forms that are BCS-1, that are highly soluble, highly permeable. The EMA also enables biowaivers for class 3 compounds as highly soluble, low permeability. So this regulatory pathway confers significant savings in time and money, specifically for generic companies when it can waive clinical bioequivalence studies, particularly in cases where the bioequivalence studies are challenging or require special patient populations. So in one of the talks this morning with quetiapine, it was mentioned that for CNS compounds, there can often be adverse drug events. So also for things like cytotoxic drugs, which require special patient populations, all of those studies can be waived with a biowaiver, assuming that your compound meets the criteria. And so I want to take a minute to just review the fundamentals behind this. I know this was largely covered in the presentations this morning, but a lot of the clients that I deal with seem to think that biowaivers are too good to be true. But when you break it down into the fundamental principles, it's clear that this is a logical and very sensible approach, and it makes a lot of sense. So the primary determinants of oral absorption are gastric dissolution, aqueous solubility, and intestinal permeability. If your formulation is rapidly dissolving in relation to gastric emptying time, then gastric dissolution is no longer rate limiting. And if your API has high aqueous solubility over a physiologically relevant pH range, which the FDA defines as pH 1 to 7.5, solubility is no longer rate limiting. And if you have high intestinal permeability, then GI transit time is no longer rate limiting. And so under such conditions, rapid dissolution, high solubility, and high permeability, if you're an immediate release solid oral dosage form that has adequate stability, your absorption should always be complete, which FDA defines as 90% or more. And therefore, small changes in the formulation itself should have negligible impact on bioavailability. Therefore, it is unnecessary to run a clinical bioequivalence study. So there are a number of techniques that can be employed to demonstrate high permeability for BCS classification, including intestinal perfusion techniques, excised tissue methodology, but the most popular is the in vitro cell monolayers, particularly CACO2, because this is a very cost-effective method. It has the greatest dynamic range, and it's highly reproducible. So CACO2 is a human colon adenocarcinoma. It's an immortalized cell line. It's grown for 21 to 28 days, and it forms polarized monolayers. So a few of the speakers this morning mentioned the transwell apparatus, which is on the right side. I don't have the pointer here, but for the actual permeability experiment, compound is dosed on the apical side of the cell monolayer and sampled from the basolateral side at various time points to calculate an apparent permeability or PAP value. And this assay is performed in both directions, A to B and B to A. The ratio of B to A over A to B gives us the efflux ratio, and that indicates the level of symmetry in the permeability. So if the efflux ratio is close to 1, that would indicate a passive transport mechanism, so no active uptake or efflux. This illustration at the top of the slide here just highlights the various pathways by which a compound may traverse an epithelial monolayer like CaCO2. So through the cells via the transcellular pathway, between cells via the paracellular route, or mediated by uptake or efflux transporters. So efflux transporters would pump things back into the apical compartment. It's important to recognize that CaCO2 and other in vitro test systems express the most common intestinal efflux and uptake transporters and enzymes that would be phenotypically relevant for the human intestine. So there are current initiatives to expand BCS biowaivers beyond class 1, particularly for BCS3 and some BCS2 compounds that are weak acids. But today what I'd like to focus on is really asking the question, are we realizing the full potential within BCS1? And what are the next steps regarding permeability, solubility, and general awareness for BCS biowaivers? So progress has been made in the identification of more accurate internal standards for permeability classification. So in order to qualify as highly permeable, a compound must be co-administered with a high permeability internal standard that has 90% or more fraction absorbed in humans, in the CaCO2 cells or in whatever intestinal permeability technique is employed. So this graph, which Sid also showed this morning, features CaCO2 apical to basolateral permeability versus human fraction absorbed or bioavailability. So this is actual human data from the approved label or published clinical data. And it plots the correlation, the in vitro versus in vivo permeability classification. So the FDA guidance prescribes the use of high permeability standards such as antipyrine or metoprolol, but the problem with those is that their permeability is essentially at the 90th percentile of these compounds. So it's possible and quite probable to obtain false negatives when using these standards. With minoxidil or other high permeability standards, you're able to achieve enhanced sensitivity and reduce the number of false negatives. Progress has also been made in the selection of optimal pH conditions for permeability testing. So a compound may be absorbed across various segments of the GI tract or along the complete GI tract, and this is expressed as local versus average permeability. So according to the FDA guidance, since complete absorption is based upon 90% or more fraction absorbed, this is more in line with the notion of average permeability. And therefore, using a single point permeability determination, you could potentially misclassify a compound's permeability. So for example, sodalol, when it's compared to metoprolol, appears to have low permeability, although the compound does exhibit complete absorption in vivo. And in fact, its permeability is completely pH dependent, and its absorption in the distal colon compensates for its low permeability in the proximal GI tract. So recognizing that local permeability classifications using optimized pH can provide a more accurate permeability classification is progress with BCS classification. However, a number of limitations still exist, and perhaps the most prominent one is the current prerequisite for passive permeability. So in order to qualify for a biowaiver, the FDA says that a compound must exhibit passive permeability. It cannot be a substrate for an uptake or efflux transporter. And so the guidance states that passive permeability can be established by either clinical PK data showing dose proportionality or in vitro lack of dose dependence. So here's an example of typical CaCO2 permeability data. For these BCS permeability classification studies, generally 3 concentrations are evaluated per the FDA guidance: 1, 10, and 100% of the highest dose strength dissolved in 250 milliliters. So permeability higher than a co-dosed internal standard minoxidil would indicate high permeability, and efflux ratio less than 3, in this case, would be indicative of passive transport. So this data would not call for any additional clinical data showing dose linearity, and it confirms the sufficiency of the test system for the BCS biowaiver. An alternate data set here indicates an efflux ratio greater than 3 at the lowest concentration. And of course, any active transport would be most pronounced at the lower concentrations. It could be masked at higher concentrations where the transporter proteins are saturated. So if this type of data were observed in this scenario, our first response would likely be to evaluate alternate pH conditions. So revisiting this and determining whether an optimal pH was used based on the compound's pKa. Additionally, the FDA guidance allows for clinical exceptions. So in cases where in vitro efflux is observed, if there is clinical dose proportionality data, that could supersede the in vitro data. So you could also apply a chemical efflux inhibitor in the in vitro system to abolish any efflux mechanisms and evaluate intrinsic permeability potential. So really there's been significant changing perspectives on active transport. And one of the reasons that this is so antiquated is because a lot of the transporter research has been done in the past 5 to 10 years. And the FDA BCS guidance was issued again in 1996 and updated in 2000. And the guidance itself acknowledges that at the time it was published, a criterion could not be set for efflux ratio or for active transport, and that is why it limits the use of BCS biowaivers to passively transported compounds. But since then, in 2005, for example, Les Bennett published stating that there's no significant transporter effects for highly permeable compounds. And then from 2006 to 2012, various regulatory agencies have published guidances prescribing techniques for evaluating transporter interactions using in vitro test systems. And in fact, these are so reliable that negative results from these in vitro transporter evaluations preclude the need for clinical testing. And these guidances furthermore state that for BCS1 compounds, transporter interactions are negligible, and therefore clinical drug-drug interaction studies with PGP and BCRP, 2 of the intestinal efflux transporters, can be waived for highly permeable compounds. Part of the rationale for this is that it is well recognized that in vitro test systems tend to exaggerate efflux. CACO2, for example, has tighter intercellular junctions and lower tight junction porosity than the intestinal epithelium. So this, as you can see here, there's about 5 to 6-fold higher efflux ratio of digoxin in CACO2 versus human intestinal tissue using an ex vivo technique. And so if passive permeability is much higher than carrier-mediated flux, then passive permeability is essentially equal to the total effective flux. And so if we revisit this case study where efflux ratio was above 3 at a single concentration, it begs the question, does this efflux even matter if the permeability is sufficiently high? To take it a step further, if you were to observe in vitro efflux ratio above 3 at all concentrations tested and high permeability at just the mid and high concentrations, should this not be sufficient to demonstrate high permeability and be eligible for a biowaiver, especially because the FDA's criteria for complete absorption is 90%. So would it not be more relevant to base the permeability on the 10% or 100% concentration? Is the 1% dose even relevant? when it comes to BCS solubility classification, there are also modifications that could be made to extend applicability for other compounds. So BCS solubility classification is a threshold-based measurement. In order to meet the criteria, a compound, the highest dose strength of the drug product must be dissolved in 250 milliliters or less from pH 1 to 7.5. So the pH range is physiologically relevant to oral absorption. Temperature is run under physiologically relevant conditions. But the BCS guidance calls for equilibrium or maximum solubility determination. And so I've often confronted cases where a compound may not be stable for 24 hours, but it could potentially be stable for the course of oral absorption. If the Tmax is 1 hour and the mean intestinal residence time is 3 hours, is 24 hours really relevant to oral absorption? And so here's a case where the solubility threshold would have been 0.2 mg per mil. The compound fails to meet the criteria at 24 hours, but at a more relevant time frame may have met the conditions. Similarly, the pH criteria, pH 1 to 7.5, again, if your Tmax is 1 hour, is your compound ever going to encounter pH 7.5 or the pH conditions relevant to the very distal GI tract? And so I mentioned that BCS biowaivers remain underutilized, and it's time. Okay, just a few quick points. I interface on a daily level with generic companies, and a lot of them perceive BCS biowaivers as a risk, as a competitive disadvantage. One of the reasons I think that could be is this recent FDA RTR guidance refused to receive standards, and the language is somewhat vague for under what circumstances they would refuse to receive a biowaiver application. I think that calls for revisiting. And also international harmonization. So biowaivers are accepted across various international agencies, but there are some differences. I mentioned EMA allows for biowaivers for class 3 compounds. The solubility requirements are slightly different. Also, EMA tends to view in vitro data as complementary to in vivo data. So striving for a greater international harmonization could also facilitate greater acceptance and use of biowaivers. So just to summarize, the main points for extending applicability would be efflux substrates, updating the guidance according to current transporter science, solubility conditions, specifically pH and time course, and spreading greater awareness and international harmonization. Thank you. I have heard that before, but technically, no, that would not be acceptable because the solubility and permeability have to be intrinsic properties of the API itself.